There are two more macromolecule families that we need to discuss. First is proteins. Take a quick look at the spelling. And then also nucleic acids. If you look at the proteins, they are maybe the best example of a chain-like macromolecule that we have. And if you take a look at them, it's actually indicated in the name that they commonly use for these, which is polypeptide. So the poly implies that there is a chain of many molecules, just like polymer when we've talked in the past. And then peptide is a reference to the bond that forms between the monomers, which is just kind of a special kind of covalent bond. That's rather unique. The monomers of these are amino acid. And amino acids are fairly unique in their own right. They have several components to them that come together to make the individual monomers for the whole protein fairly unique. And so there's actually different levels of structure that we talk about in protein. If we start at the most simplistic, which is what does a single amino acid look like, there's 5 parts to an amino acid. There's a central carbon, and attached to that carbon there's going to be a hydrogen, always, a carboxyl group, which remember is a COOH group. And those COOH groups make things acidic. So that's where it gets the name acid as an amino acid. And then there's an amine group. That's why we refer to these as amino acids. Amine groups are a nitrogen with 2 hydrogens attached, an NH2. And then lastly, you have a functional group. And the functional group varies between amino acids. There are 20 different functional groups and 20 different amino acids. And so we represent the functional group with the letter R. If you think about math where they use the letters X and Y as variables, and you can plug different things in for X and Y and get different answers, the functional group is very much like that it gives each amino acid its unique properties, because all the other parts, the carbon, the hydrogen, the carboxyl group, and the amine group are the same across all those amino acids. It's just this one add-on that gives each amino acid those unique properties. And some of those properties include things like acidity, things like alkalinity, being hydrophobic, being hydrophilic, aromatic properties, meaning they smell, things like that. One way that I have come to think of amino acids and help me remember those pieces and kind of their roles is a really kind of silly visual analogy, I guess. Thinking about the idea that each of them has somewhat unique properties. Pardon me, I did some kind of roughshod drawing there. And so I've started thinking of amino acids as a popular toy, which I'll draw out for you here. So remember that the base of an amino acid is that central carbon. So here's my carbon, and I'm going to turn it on its side to serve my purposes here. That's a big letter C to represent that central carbon. And I'm going to build off of this for the rest of it. So the other thing that all amino acids have is hydrogen, which I'm going to put up here as a little hat on my structure here. They have that amine group. I'm going to put that down here as NH2. And we've got that carboxyl group, COOH. And we've got that R group that gives each its unique properties. So I'm going to put the R right here. Looks to me like Mr. Potato Head. I don't know about you. But this is a kind of strange Mr. Potato Head because he only has one hat, and it's always the same hat. He only has one pair of eyeballs, and it's always the same. He only has one pair of feet, NH2 for 2 feet. Fits kind of nicely there. And so there's a lot of things about this Mr. Potato Head that can't change, but I can change this mouth because this functional group can vary. So if I put big lips on my potato head, it becomes Mrs. Potato Head. If I put a pipe in my potato head, it's Professor Potato Head. If I put bucktooth in my potato head, now it's Redneck Potato Head, and so forth. And so just by changing that one thing, that functional group, I can change how you perceive the entire structure, even though I've only changed one thing. And amino acids work in that same way. Okay, if we take a look at the functions of proteins, the functions are quite varied because we have all these different kinds of amino acids, and we're going to make chains of those amino acids, and there's almost an unlimited variety of ways in which those chains could be assembled. So proteins have tons and tons of variety because the chains can be different length, they can be different shapes, they can be different arrangements of those amino acids, and so the possibilities are practically limitless. Particular functions that are common include structural functions, often involved also in movement, sometimes acting as chemical messengers, and lastly as enzymes. And some ways in which you've probably seen these are, for example, on a large scale, hair and tendons in your joints are good examples. But then also, for example, on a small scale cellular level, cell identification is carried out by proteins, and cell adhesion is carried out by proteins. So how cells stick together to form larger structures. In terms of movement, you guys know your muscles are heavily involved in how you move, and most of what a muscle is protein. That's why when you eat muscle-based foods like steak and pork chops, you get lots of protein from them because that's what muscles are made of. On a smaller scale, there's transportation in cells where proteins act like little conveyor belts that substances can be pushed along or pulled along to move them throughout the cell. In terms of being chemical messengers, a lot of hormones are proteins, as are a lot of neurotransmitters used by the nervous systems. And lastly, for enzymes, these are going to be used to speed up chemical reactions. Elaborate more on that in the chemistry and reactions section. But remember, chemistry is kind of an inherently slow thing, so enzymes' ability to speed it up to the speed of life is actually quite critical. So lots of variety in function because there's lots of variety in structure. On top of all the different amino acids and the fact that these are chains of amino acids, there are additional levels of structure. So there's 4 levels of structure we talk about. I would like you to know what it is and also what the bonding type is within that level. So the first level is referred to as primary, and we often use a 1 with a circle. How they denote 1 prime in math. And what that level is just a description of the order of the amino acids. So the simplest way to describe a protein is to just list off the name of the amino acids in the chain. For example, there might be a lysine and an isoleucine and a glycine and a tyrosine, and just what order are they and what's there. These again are connected by those peptide bonds which is a special type of covalent bond. The secondary level is that depending on the order of amino acids, you will often get twists and folds. And those twists and folds are held together through hydrogen bonding. Remember, those aren't really bonds, they're actually just these weak kind of magnetic attractions between the molecules. And so it's not a very sturdy level of structure. So if we're going to maintain that, we need to support it some way. And that comes in with the tertiary level, where there is a reinforcement of the secondary level. That's carried out by disulfide bonds. Now you'll notice if you look back that we haven't talked about sulfur in carbohydrates. We haven't talked about sulfur in lipids. We aren't going to talk about it in the nucleic acids. The one place where sulfur is really critical is in the structure of proteins. And proteins cannot hold their shape without disulfide bonds. And shape is very critical in chemistry, so the sulfur is essential in this role. Last level of structure is called the quaternary level. And the quaternary level involves the joining of multiple chains. So you build one primary, secondary, tertiary level, you build another one, you build another one, you build another one, and they come together. It's a combination of covalent and ionic bonding, depending on what's in those previous levels of structure. And it really gives you more possibilities in what you can make and what shapes you can create, because you have this kind of set group of building blocks, if you will, but you can make so many different things from them by joining them together. My kids always want new Legos. They go, why do you want new Legos? Well, because this Lego set will build me a Death Star from Star Wars, and that one will build me, the SpongeBob Pineapple House, and that one will make me this. But what happens more often than not is that all those unique seeming sets come together and make things that, you couldn't even imagine, because it's not what they were meant to be, but they can be joined together in these new, unique ways. Now, this whole levels of structure thing sounds really complicated, but if you think about it, we use levels of structure all the time. In particular, we use it in language. That at the end of the day, our language has levels of structure, where first you might learn your letters, and then you learn to form words from them. And then you reinforce what those words mean by joining them together into sentences. And then those sentences can come together, each independent sentence, to tell you a story. I think that's a really nice analogy for understanding levels of structure and how levels of structure can enhance the most fundamental aspects of something, like the fact that at the end of the day, the English language is just a bunch of strings of letters. So hopefully that'll help you a little bit. Here's some pictures that also might help. We've got primary structure, just what are the amino acids. You don't need to know the names of any of the amino acids at this point, and probably not at all, to be honest, for this class. But we often represent them with these little three-letter abbreviations like leucine and glycine and alanine. And then the relationships between the chemistry of some of these will draw molecules towards each other, very much along the lines of that hydrogen bonding we discussed. And so as it does that, it will pull them into shapes like what they call a helix or a twist or a fold. And then in order to keep that shape, these disulfide bonds, depicted here as these little discs, reinforce and support what was created by the hydrogen bonding. And then lastly, you can pull several of those structures together and create a full-blown protein that has, you know, lots of complexity and can carry out large functions. Because proteins are so based in shape and there's all these layers of structure there, they are susceptible to something called denaturation. This is basically that the chains can unfold if conditions change. And the most common changes that influence protein shape is changes in temperature and changes in pH. And it's not real clear cut. Sometimes that's heating, sometimes that's cooling, sometimes it's acid, sometimes it's alkaline conditions. And so each protein has kind of its unique scenarios under which it might unfold. But you guys see this often. For example, if you take an acid and add it to something like milk, you'll find that it will curdle the milk, make it clump up and chunk up. And that's because the acid has unfolded the proteins and turns them into kind of a solid. Another good scenario is heat added to an egg, which a lot of people are aware is a good source of protein, will solidify that egg. Whether it's a hard-boiled egg or a scrambled egg or a fried egg, doesn't really matter, but it does that. And so your body and living systems can use this property to control the action of proteins. For example, the proteins in your mouth are not going to behave in the same way when they get to your stomach, where the pH has changed as well as the temperature. And the enzymes and proteins that are active in your stomach are going to change behavior as they move into the intestines and get buffered and are no longer under the influence of your stomach acid. So this is really useful to be able to not only create proteins with all these great functions, but then to be able to control them by influencing their environment. Last macromolecule is the nucleic acids. You will sometimes see the term nucleotide and nucleoside in reference to these. The monomers are called nucleotides. And there's going to be 3 parts to a nucleotide. There's going to be a pentose sugar. There's going to be a phosphate. And these two pieces together are very often referred to as the sugar phosphate backbone. So you might see that. And then lastly, there's going to be nitrogenous bases. Nitrogenous just means nitrogen containing, and they don't mean bases as in alkalis, they just mean as in pieces. There are 5 of them, 4 you're probably somewhat familiar with from the abbreviations we use. We usually use the letters A, C, T, and G for the 4 most common ones, and those represent adenine, cytosine, guanine, and thymine. And the last one, which is less common, is uracil. We don't hear much about uracil because it's only found in RNA, and we very rarely talk about RNA. A lot of people are familiar with DNA, but haven't heard as much from the media about RNA and how it works. So here's some of those structures. Here you see what a phosphate group looks like. Not something I'll ask you about, but those are just for those of you who are curious. Here's one of those pentose sugars. And here's what some of these nitrogenous bases look like. All circular ring structures, kind of interesting. Functionally, I think looking at these kind of side by side for the DNA and RNA is going to be helpful. There are some other representatives of this group, but let's talk about the DNA and RNA first. And we're going to do a little bit of both here, not only function, but also what makes them unique structurally. So let's look at the structural parts first. The pentose sugars for DNA is, of course, deoxyribose. And the pentose sugar for RNA is, of course, ribose. DNA, as I mentioned, uses adenine, cytosine, thymine, and guanine. RNA uses adenine, cytosine, uracil, and guanine. So a slight variation in which bases they use. DNA utilizes those to create a double-stranded helix. A helix is just a twist. And so most of us have an image in our head that DNA looks something along these lines. RNA, on the other hand, is a single strand, so it can't form that helical shape. DNA's purpose is storage. Storage of instructions to make proteins. And remember, proteins are variable and complex. So we need an entire recipe book just to figure out how to make the proteins we need. And there's literally hundreds of thousands of different kinds of proteins being used by your body. RNA is more for the transport of those instructions to make proteins. Because DNA is all about storage, it is only found in the nucleus and is protected carefully in that nucleus, whereas RNA is going to be throughout the cell because the structures where the proteins are actually made are not in the nucleus. And the reason you want to keep the DNA safely in the nutrients as a storage point is that they are safer there. And they're safer there and need to be held there in a safe place because DNA is permanent. If you alter or change that DNA, those changes become part of the instructions now for making proteins. Whereas RNA is recyclable and reused. It is assembled and then disassembled to suit the needs of what proteins we need in this particular moment. So DNA and RNA are all about information, this information to make proteins. The other representatives of this group are about energy, which, as much as information is kind of an abstract concept, you can't hold information, you can't transport information really, it's an abstract thing. Energy is in a lot of ways the same thing. Remember, energy is not matter. So how do you hold it? How do you move it? How do you do things to it? And so the other nucleic acid representatives are going to handle energy. The representative of that family that we're going to talk about that manages energy is adenosine triphosphate. And so what adenosine triphosphate is a ribose and an adenine and 3 phosphate groups. And what adenosine triphosphate, or ATP, is going to do is it holds the energy in the bond between the second and third phosphate. If it loses or transfers that energy, that third phosphate drops off and this thing becomes a new molecule, ADP, or adenosine diphosphate. So this is not a real permanent molecule. This is a molecule that's in transition depending on the amount of energy that it's trying to hold. This is an image I got of this one. So this is in its charged form, 1, 2, 3 phosphates on here. But this is where that energy is held, is what in the bond between the second and third phosphates. Yes, I know this image is in French, but I really liked it. And so a lot of times we think of it like a rechargeable battery, where when you've got all 3 ATPs on there, it's charged. And as it releases the energy, one of those phosphate groups falls off and it becomes its ADP form. This is often referred to as the ATP cycle, which I'm just going to superimpose on here. So we'll put ATP up here and draw a big circle around this image. And I'm going to put ADP down here. That's the low energy form. And where does ATP let energy off? Well, it's for cells when they need to do work. So anytime that a cell needs energy to carry out a chemical reaction or to move something or to create an impulse, it uses the energy from ATP. And as it does that, it's going to let off that phosphate group. Now you've got ADP. And when we gain new energy that we harvest from food, we're going to grab a phosphate from somewhere out in the cell or out in the surroundings and attach that back on to get us back to having ATP again. It's a fairly simple cycle. But so many of the things we do require at least some small amount of energy or let off some small amount of energy. And so we constantly are using ATP to hold that energy and move it to the places where we need it the most.